Hi, everybody, and welcome to Echo Live. My name is Anna, coming to you from the Michigan Science Center's Echo Distance Learning Program. Today, we are talking about Genetics 101, which, again, if you've been tuning into Echo Live, biology is my background, so it's something that I've got a lot to say about. But we've also talked about genetics a few times on Echo Live so far. So I've got some questions for you to test out your background knowledge about genetics and what we've learned so far throughout our time together on Echo Live. Now, if you haven't done so already, go ahead and introduce yourself in the chat feature of whichever program you are tuning in from. I can see we have friends joining us on Facebook, on YouTube, and right here in Zoom. Hello, everybody, and welcome. If you haven't done so already, introduce yourself in the chat so that I know you're here, know you're watching, and know that you're able to use that chat feature to communicate with me throughout the program. I can see Steve tuning in. Hello. Helena is excited about biology. Maybe some future biologists over there in your house. I know that it is absolutely one of my favorite things to talk about. So I'm so glad to see that you guys are enjoying this and loving biology just as much as I do. So once you've introduced yourself in the chat, the first question I want to ask you is, does anyone remember from a past episode of Echo, or maybe because you know this on your own, what do we call the genetic material that encodes everything that is a living thing? So what do we call the genetic material that makes a human, or a banana, or a chicken, or a tuna? What do we call that genetic material? It's something we have mentioned here on Echo Live, but it's something you might have also learned about on your own. What do we call that genetic material? It's packed inside our cells, and it's what makes all of us exactly the way we are. We're all unique in our own ways, and it's all thanks to this really, really important piece of genetic material. Some friends starting to send it in. That's exactly right. We call it DNA, or more officially, we call it deoxyribonucleic acids. But wow, is that a mouthful? So for the purposes of this episode of Echo Live, we'll stick to calling it DNA because most scientists do. But DNA is what makes us us. It's what makes a human, a banana, and a chicken, right? But DNA is all encompassing, and it encodes a lot of information. Did you know that about 60% of human DNA is exactly identical to bananas? So a lot of DNA goes into just making a living thing living. But let's talk a little bit more about DNA and what it looks like. We mentioned DNA most recently during our crystals episode of Echo Live, where we talked about how a very notable female scientist named Rosalind Franklin actually took the first photograph of DNA using what we call X-ray crystallography. It was a revolutionary photo because it confirmed what DNA actually looked like, because it is much too small for us to even see with a microscope. We can't even see it with the microscope. It is so small. But let's take a look at what DNA would look like. And first, we'll take a look at it in the form of a drawing or an illustration. So over here, let's first take a look at this image. This image on the left side of our screen is an illustration of a DNA molecule. We call this twisty ladder shape a double helix. So two strands wrapped around each other into a double helix. This structure was only confirmed to be possible because of that image that Rosalind Franklin took using X-ray crystallography. But Watson and Crick also contributed to this DNA structure because they figured out what we call base pairing. Now, on each molecule of DNA, we have two long strands, which we call sugar phosphate backbones. So these long blue ribbon-like structures in this illustration, those are what we call the sugar phosphate backbone. There are two of them, and they are linked together by hydrogen bonds between nucleotides. Now, I know this is turning into quite the vocabulary lesson, but bear with me because this is really important to understanding how DNA encodes information. Now, nucleotides, these colorful pieces that make up the rungs of the ladder, those are what encode information. And they can arrange themselves in different orders depending on what type of organism or species you are looking at. But they fit together like puzzle pieces. So you'll notice that if you look at this drawing, adenine, the green nucleotide, only ever pairs with thymine, that red nucleotide. In this image, they did a good job of noting that in their shape. You can see that the green point on adenine would fit right into thymine just like a puzzle piece. But adenine can never pair with cytosine. You'll notice that this one has a rounded opening instead of a pointed opening. And cytosine can only ever pair to guanine. So cytosine pairs to guanine, adenine pairs to thymine, and there is no other possible combination of these nucleotides. If we take a look at the image on the right, we can tell exactly why that is. It's not actually because they're pointed or rounded. It's because of the number of hydrogen bonds that connect those two nucleotides and form what we call a base pair. So between adenine and thymine, there are 2 hydrogen bonds, and they line up just perfectly. If we were to flip one over or change it out, we couldn't make those hydrogen bonds. So adenine pairs to thymine with 2 hydrogen bonds, but guanine pairs to cytosine with 3 hydrogen bonds. So 2 between A and T and 3 between G and C. And the way I like to remember my base pairs is that G pairs with C because of Girl Scout cookies. That was taught to me by my 9th grade biology teacher, Mr. Bechtel. So shout out Mr. Bechtel for teaching us that guanine and cytosine pair because of Girl Scout cookies. Just a little trick that I like to use to remember it. But if you are looking for a way to examine this 3D double helical structure of DNA at home, you can recreate this using some really delicious edible materials. So on my paper plate here, I have some licorice sticks. Those are going to act as our sugar phosphate backbone. So those blue ribbons in our previous illustration are represented by these 2 licorice sticks. And then to act as my nucleotides, I have a whole bunch of these colorful mini marshmallows. And these actually work perfectly because when you buy these colorful mini marshmallows from the store, they are flavored, by the way, they actually come in 4 different colors. So it's as if they were designed for this exact experiment. We have green, yellow, orange, and red. And so you can designate each one to be a specific nucleotide. So we could say that green is adenine, yellow is thymine, pink is guanine, and orange is cytosine. And I know it's not super easy to tell, but if you were to do this at home, you could tell much more easily the colors of our little mini marshmallows here. So remember that pink and orange can only pair with each other, and green and yellow can only pair with each other. As you are constructing your DNA molecule, keep your base pairs in mind because that is super, super important when it comes to this structure of DNA. There's also another interesting fact that they fit because of those bond sites, but also because some are much larger than others. So if you tried to pair an adenine with a cytosine, it might be too large for the backbone. On the other hand, it might be too small for the backbone. So nature really laid this out perfectly, just like a puzzle. So the way we're going to construct this is we're going to be attaching our base pairs together with toothpicks. These are going to act as our hydrogen bonds. And so you can pick these arbitrarily. We're not worried too much about what information we are encoding just yet with our base pairs, but remember that there's a certain pairing order that you have to follow. So green and yellow, orange and pink. Do another, we'll do pink and orange. And you'll notice that I'm leaving just a little bit sticking out so that we can still also connect them to our sugar phosphate backbone. Let's do 2 more. So another green and yellow. And then one lastly, let's do another yellow and a green. All right, so we have our base pairs set and they are paired appropriately. And then we said that the object of base pairs, right, is to connect the 2 sides of our sugar phosphate backbones together And once we do this, so we'll set them up just like we said, like the rungs of a ladder almost. So we'll poke all of our base pairs formed by those hydrogen bonds between our nucleotides. We'll poke those into one of our sugar phosphate backbones, and then we'll just connect the opposite end. But the important thing to notice once you set this whole thing up is that right now, we're not actually forming a helix. So we're not forming a helix just yet. You are going to form the helix because once you have your base pairs connecting your two sides of your DNA structure, if you want to be able to see this in 3D, you're going to have to take the two top and bottom nucleotides and twist them into that three-dimensional structure. And so this structure, like we said, is what we call a double helix. They coil around themselves as those base pairs are formed with hydrogen bonds. And so this now looks much more similar to our illustration that we saw in the previous slide. And the best part, of course, is that everything on here totally edible and delicious except for the toothpicks. So please don't eat the toothpicks. Your hydrogen bonds, not very tasty, but mini marshmallows and licorice, both delicious. But if you can think of other materials to make your own DNA model with, send us a picture. I would love to see your model of DNA, and you could use anything. I like to do this because of those 4-colored mini marshmallows, but you can really use any sort of materials that give you this same DNA double helix structure, which is very, very iconic. And again, thanks to Rosalind Franklin, one of our favorite female scientists. All right, so next let's talk a little bit about what genes do, right? Now, humans, we said, have about 60% of the same DNA as a banana, which is crazy to think about, right? But there's a lot of DNA that's basically junk. Not to say it's not important and we don't need it, but it's not necessarily what gives us our different diversity and our different human traits. Between humans, there's even less variation. So between any two humans living here on Earth, we are about 99.9% of the exact same identical DNA. Any two humans, anywhere in the world, share 99.9% of their DNA. That's only 0.1% difference. And that's what gives us our different traits, right? It's what makes you look different than your brothers or sisters. It's what makes you look different from your neighbors. But it's also, again, like we said, those big differences are what make humans humans and bananas bananas. But DNA is really small, right? So those differences between us are minuscule. Now, to understand just how small DNA is, right, we said it's so small that we can't even take a picture of it under a microscope. What we can see is kind of a big clump of it. So if we look right here on my background, this big dark clump in the center of these cells, if you didn't know, these behind me, these are human cells that are going through division. This big dark clump there in the center, that is all of that cell's DNA compacted into one tiny little piece of a cell that we call the nucleus. So if we take a look at the nucleus of a cell, it is this dark spot, usually very centrally located, but it's always a big dark spot because it is so full of DNA. All of the genetic material inside any living cell is contained inside that nucleus. It's there to keep it safe, and so that DNA can do its job and code for all the different life processes and jobs of the cells that they are a part of. Now, even though DNA is so tiny that we can't see it with a microscope, and we certainly can't see it with our naked eye, it's actually incredibly long. If you were to unwind all of that genetic material inside your cells, each strand of DNA is about 1.8 meters long. 1.8 meters long. That is crazy. That's over 5 feet. That's actually almost 6 feet in length. So what I have here, which you might not even be able to see, is a piece of sewing thread. And this sewing thread is 5 feet 10 inches long, which is almost approximately the exact same length as my human wingspan. Fun fact that your wingspan is almost identical to your height. And since I am almost 5 foot 10 inches tall, I have it stretched all the way across me, this long piece of sewing thread, but you probably can't see it. Even though this strand, right, encodes for everything that makes us us. What you can see is if I were to take our thread and coil it up really, really tightly around my finger, we'll be able to see it much more easily. And that's exactly how DNA exists inside all living cells, is coiled up inside that nucleus. So if I coil the DNA here around my finger and then we take a closer look, now we can see that string wrapped around my finger here. So DNA exists just like this. When it's coiled up and packaged really, really neatly and ready to be used to encode information, that is what we call a chromosome. And humans have 23 copies of 23 chromosomes and 2 copies of each one. So these are 2 normal human karyotypes. Normal in genetics doesn't necessarily mean normal by the traditional sense, but in terms of genetics, we would call this a normal male karyotype and a normal female karyotype. Each little pair of these 2 little kind of squiggly lines are 2 chromosomes. So we have 22 regular chromosomes, and then we have our sex chromosomes, which are what makes a genetic male different from a genetic female. We get 1 chromosome from each parent organism. So one parent A gives us 1 chromosome, so maybe this half of number 6, and parent B gives us this half of chromosome 6. Same things go for our sex chromosomes. We get 1 from parent A and 1 from parent B. If you get an X and a Y chromosome, notice how different they look. That makes a genetic male. If you get an X from parent A and an X from parent B, notice that there's no shorter Y chromosome over here. 2 X chromosomes make a genetic female. But there are, of course, what we call mutations. Mutation is what gives us all of the diversity of life here on Earth. Life is constantly changing, and genetic mutations have given us a lot of adaptive benefits over thousands and thousands of years. It's what allows us to grow strong crops, and it's also sometimes what accounts for differences between people as well. Now we are going to perform an extraction of DNA, of those chromosomes, from a living organism. So our living organism that we are going to be extracting DNA from for the purposes of this lab is not from a human, but it's from strawberries. And the reason we use strawberries for this DNA extraction lab is because of how much DNA is packed into each of their cells. So humans, we said, have 23 chromosomes. We get 1 side from each parent, so 23 chromosomes, but strawberries get actually 8 copies of each one. So in humans, we call us diploid or diploid, meaning we get 2 copies, 1 from each parent. But strawberries get 4 copies from each parent for a grand total of 8 of every single one of their chromosomes. That means their cells are just absolutely bursting with DNA, which make them fantastic for this extraction. So we need a few things in order to get this DNA out of these strawberry cells so that we are actually able to see it, which is going to be crazy. What we're going to do first is we're going to take our strawberries, we're going to remove our stems here because we don't want the leaves. We're just looking for the cells, the healthy red cells of our strawberries here that are full of all that genetic information. Take off our leaves, and we're going to drop them down inside a Ziploc bag. And then one of my favorite science words I've learned from doing this extraction is we are now going to pulverize our strawberries. And by pulverize, you might know what I mean. I'm going to smash them into smithereens. So as I smash up the strawberries, right, I am already starting that process of breaking some of these strawberries cells open. But we need a couple other ingredients to truly get that DNA out of those nuclei, out of those really, really centrally located parts of our cells, like you see in this picture behind me. So we're going to add some dish soap next. Now, we've talked about dish soap on Echo Live in the past too, mainly about its role as an emulsifier. Its role as an emulsifier means that it's actually able to penetrate the cell membranes of these strawberry cells. So both the outside barrier of our cell, like in this image behind me, and the barrier that surrounds the nucleus. The soap is going to break apart those cell walls and infiltrate it. So it's actually going to be the part that releases the DNA from the cells. We're also going to add a pinch of salt. Salt, we've talked about, can interfere with those hydrogen bonds that we know are formed between the nucleotides. So just a pinch here, just a little bit, is also going to help that process of having our DNA strands be released and then also be soluble in our water, which we're pouring in next. So the three things we've added to our mixture here, besides our strawberries, are the dish soap, which breaks open the cell walls, our soap, which interferes with those base pair bonds, hydrogen bonds, and then some water, which is going to actually dissolve our DNA, because DNA is soluble in water, which is important to life, and it'll be important as we go through this experiment as well. Now, as you pulverize your strawberries and really get your mixture homogenized, right? We've talked about a homogeneous mixture is one that's uniform. So we really want to mash everything together, really release all of those DNA strands into our solution here. But we're trying not to form bubbles because we want the dish soap to act as that emulsifier and not be tied up forming bubbles. You'll notice that it's impossible to avoid forming some bubbles. So it's okay to have bubbles. But again, making bubbles is not the point of this experiment. So we'll really try to mash up some of those bigger bits of our strawberries here, really pulverizing, releasing that genetic material, deoxyribonucleic acid, into our solution. Now, we want to strain our solution here. So I have a mesh strainer, what in science we might call a sieve. And I'm just going to pour my pulverized strawberry solution through that sieve. So slowly, that sieve will do its job and catch all of those large strawberry particles that did not get pulverized and have not released their genetic information. And then what we're left behind with is this solution that is just packed full of DNA. But we still can't see it, right? Because we said that DNA is soluble in water. So those DNA strands, even though they've been released from their cell walls, are floating around in this solution, and they are not easy to see. So what we're going to do is I'm going to take off my strainer, set that off to the side, and I'm going to pour our solution down into this glass test tube. Carefully pouring it in, remembering that there is dish soap in this, right? So we're trying to avoid as best we can forming bubbles in our solution. So the best way to do this is by pouring it down the edge of the glass. All right, I did a pretty good job. There are some bubbles on there. But lastly, what we need is something that is not going to allow the DNA to dissolve. So even though DNA is soluble in water or H2O, it is insoluble in rubbing alcohol or isopropyl alcohol. So we're going to go ahead and I'm going to set this back down inside my beaker there. And we're going to do the same thing with our isopropyl alcohol that we did with our strawberry mixture. I'm going to very carefully and gently pour it down the side of our test tube glass. We don't want to shoot the isopropyl down into the mixture. We actually want to form a layer of isopropyl alcohol on the surface of our solution. Since the DNA is much lighter than the solution down here, since it's much less dense, it's going to float up into our isopropyl alcohol, but it will not dissolve. It's actually going to clump together, which is going to allow us to see it. So remember, we're pouring this in there very gently, trying to avoid having our isopropyl alcohol mix with our solution, but just form a floating layer on top. And you'll be able to see that a bit better once I pull my test tube out of the glass, very, very gently. All right, now our process is starting to work. Now, what we see down here is actually not bubbles. If they were bubbles, we would see those bubbles very quickly rise up to the surface and float. But what we just saw is DNA. This is millions and millions. Let me see if I can zoom in even farther. Up here at the top, just underneath my fingers, that is millions and millions of strands of DNA, which we released from those strawberry cells by adding our dish soap and salt. Now, you could try to collect this. So if you were at home trying this experiment, you might need something a bit longer than my toothpick here, or we can add more isopropyl alcohol to get it to float all the way to the top. But what we've collected is strawberry DNA. We said it is not soluble. So you can see that as our DNA comes out of our solution and floats up into our isopropyl alcohol, it clumps together, forms these big wads of DNA. So they're tiny, but we said that's millions and millions and millions of strawberry DNA strands. And you can actually collect it on a toothpick or some other sort of coffee stirrer, and you will actually get to hold DNA, or the genetic material of strawberries, in your own hands. And they'll notice that it feels really odd and squishy, but you are actually feeling DNA in your own two fingers. Now, you can also do this with your own DNA. It's a little bit more difficult. The process is exactly the same, but if you wanted to try to visualize and touch your own DNA, you can actually take a Q-tip or a popsicle stick and scrape gently the inside of your own cheeks. So that will collect some cheek cells. And then if you go through this exact same process, which of course is linked on the Science Center's website at our at-home science page, if you go through this exact same process, you can extract your own human DNA just by following these same process. Now, remember that we said strawberries are much more full of DNA than humans are. So you might not get quite as much genetic material, especially because you don't want to scrape off enough skin, right, that it's about equal to the size of 2 strawberries. But you will be able to see your own DNA just by following this same process, because we said DNA is DNA. Whether it's a strawberry or a human, the structure is the same, but the information encoded in it is different. Now, there's one last thing I want to talk about before we head out today. And this is talking about those differences in genotypes and phenotypes. Now, genotypes are encoded by these 2 different types of traits. So on this example of what we call a Punnett Square, which is how geneticists will actually determine the probability of certain traits being passed through generations, an example of this would be talking about eye color. So on the top of our Punnett Square, these 2 genes are from parent A. These 2 genes are from parent B. So we have capital B, little b from each parent, but these are 2 different parents. Now, if we look at our table over here, or our guide, the capital B, little b would indicate brown eyes. And little b would represent blue eyes. So if we were to quickly, I know we're running out of time here, fill in our Punnett Square, we would see that if we were to follow our Punnett Squares by adding a trait from each parent into each box, so in this one, we would have capital B. In the next one over, if we took capital B from parent B and little b from parent A, we'd have big B, little b. Same thing for this one down here. We would have big B, little b. And then lastly, over here in the bottom, we would have the only representation of what might give an organism blue eyes, according to our legend here. So these Punnett Squares help us to determine what is the probability of different traits or genes being passed down through generations. But genetics is much more complicated than this Punnett Square makes it seem. Even though this could tell you the probability of whether or not your offspring would have brown eyes or blue eyes, the more we learn about genetics, the more we realize that it's just not that simple. There are actually 15 different genes that code for eye color. And you probably know someone that doesn't have blue eyes or brown eyes. They might have green eyes or hazel eyes. And some people can even have what we call heterochromia, meaning that you have 2 different eye colors. So you could have a brown eye and a blue eye, or an eye that's both brown and blue at once. Genetics are incredibly important to understanding human life, which brings us to have you ever had your DNA tested? Now, there are all sorts of commercial companies that will DNA test for you. They will actually sequence your DNA, and they can tell you all sorts of information about you on a genetic level. Of course, they can't tell you anything about your personality because we know those things are learned and not necessarily encoded by genes. But some people are really interested in finding out about different sorts of medical traits that they might share or be able to pass on to their offspring. But most commonly, people are curious about what their DNA can tell them about their heritage. And I want to give you a word of caution about using these commercially available DNA tests to tell you about your heritage or your nationality. The reason being, genetic testing is relatively new, and these different sites that you can send your DNA to, so normally it happens just like this, actually. You would actually scrape your cheeks or spit into a tube, mail it off to a lab. They would extract your DNA, turn it into millions and millions of copies, and then they actually compare it to genetic samples, which they've collected from all around the world. So all around the world, they have to have a sample pool to compare your DNA to. So these testing sites are only as accurate as the data that they've collected from known sources. So most of them have done a decent job of collecting DNA from all different regions and areas of the world. But what if they don't have DNA to compare your DNA to that is similar enough to your ancestors? We said that all humans on Earth are over 99.9% of the exact same DNA. The only things that scientists are looking for, geneticists are looking for when they do these different tests is those 0.1% differences. There's actually more genetic diversity within the continent of Africa than there is anywhere else on the Earth combined. That's to say that if you were to take a person from Southern Africa and compare them to another person from Central Africa or even Eastern Africa, their DNA would have more differences between them than if you were to compare one of those people from an Asian person or a European person or a South American human. This is all to say that race is not genetic. There is no scientific basis that explains race. Race is a cultural construct. Humans just haven't been around long enough for us to develop different subspecies. Just like eye color, all of the differences that you see between races or different people from different areas are just products of their genetic traits that were passed down to them from their parents. This happens for different reasons, right? The eye color that you see, whether you have brown eyes or blue eyes, is all to do with how much melanin is in your irises. So people with more melanin in their irises have brown eyes like I do. People with less melanin can turn out with green or blue or hazel eyes. That's the same pigment that makes skin either darker or lighter as well. More melanin makes your skin darker, less melanin makes your skin lighter, but it has nothing to do with us all being part of the human species. There are no subspecies of humans. We are all the same. We developed these, right, because melanin helps populations to defend their skin against the harmful UV rays of the sun. But those aren't the only differences between people who have descended from different parts of the world. There's studies that show the farther you are from the equator, the shorter people get. That's because it's people who are farther from the equator deal with much colder temperatures, and scientists think that having a shorter stature allows your body to keep itself warm more easily. So people who are descended from people living close to the equator are usually or historically much taller because they can be taller and still maintain their own temperature. Zara asked a great question, is why is human DNA different from other animals? And that's for the same reason. Just as humans can diversify to survive in different environments, right? Animals deal with all animals and fruits and plants. We all deal with different sorts of environments, right? We use different processes to produce or find food. We exist in different climates. Plants grow out of the ground, whereas people are mobile and walk. So there are all sorts of differences, and DNA, those puzzle pieces that connect our two sugar phosphate backgrounds, that encodes everything. It's what makes you tall, it's what makes you short, it's what makes you have different eye colors, it's what makes you have different eyes, different skin colors. Everything is encoded in DNA, and it's what makes a strawberry, right? So instead of growing legs, strawberries grow roots. That's not to say those are alternatives of each other, right? But we're all living things that face different obstacles throughout life. Now, I know I'm a little over my time, or at this point, severely over my time, but if anyone has questions about DNA or genetics or these experiments that we've tried, go ahead and type them in the chat. I'd love to answer them. Of course, at this time, we'll take just a moment to thank the sponsors of Echo Live, like Ford and Denso. They allow us to bring these free Echo Live programs to you every weekday while the Michigan Science Center is closed to the public. If you're looking for guides for some at-home science experiments, you can check out this link that's listed on the bottom of this slide here. And of course, make sure you are liking, following, and subscribing to the Michigan Science Center on all of our different social channels so that you get updated about Echo Live and all of our different various experiments that we suggest you try at home. Let's see. Kelly asked, what if everyone in your family has different eye colors? And that's normal. Now, you can actually have two brown-eyed parents that give birth to a blue-eyed child, and that goes back to our Punnett Square. Even though both of those parents, capital B, little b, even though both of those parents had brown eyes, there was still a 25% chance that their offspring could have blue eyes instead of brown. So it is totally normal to have eye colors different from everyone else in your family because everyone is being passed down traits through generations. And we said that most traits that are observable on the surface are more complicated than blue versus brown. There are 15 different genes that you would get a copy of from each parent. Some of them are dominant, like brown eyes are dominant. Some of them are recessive. And so the recessive ones can pop up much further down the line in populations as they're carried down across generations. That's a great question. Someone asked, what is the next episode? Tomorrow, we're going to talk about music making. And so we're going to teach you how to make some different musical instruments from things that you probably already have laying around your house. You're not going to want to miss it. We've got a really fun show planned for you, and we'll be teaching you how to make something at home. So it'll be really cool. I hope you'll all be back here tomorrow. And then last question before we head out. Jamie asked, will these episodes keep going when the Science Center opens back up? And the answer is yes, although they might not take place every single weekday. We want you to visit the Science Center if you are located around us. We want you to go out and enjoy your summer vacation or to participate in our virtual summer camps, which are advertised on the Michigan Science Center's website. Those will be your way to engage with us in science every single day. But Echo Live will keep going. We will still provide you episodes of Echo Live every week. We'll still teach you experiments to try at home. We'll still show you some big, exciting stuff that you can't try at home because, of course, that's really fun and exciting too. So don't worry, Echo Live is here to stay, and we just want to be a part of your summer, but we also want you to get out and have some fun and do stuff besides just hang out with me here on Echo Live. Thank you to everyone who's been tuning in so far. It's been amazing. And, of course, I hope I'll see you back here tomorrow for another episode. Again, we'll be talking about musical instruments, and so I'll see